All right, so we'll get started. So welcome everyone to the February installment of the BIRC Speaker Series. Today we're very happy to have Professor Jim Haxby here from Dartmouth. So Jim has a PhD in clinical psychology from University of Minnesota. I guess was practicing for a bit. He was the chief of the neuropsychology unit at National Institute of Aging for a time, then came over to the dark side of functional neuroimaging, where he was the section chief at NIMH during some of the early years of fMRI. His work focuses on the neurobiology of visual processing, especially face processing. And he's perhaps best known for his work on hierarchical models of face processing and the interplay between those and social cognition. So this is great news, both if you study faces and more generally in cognitive neuroscience, because visual processing is a great testbed for new neuroimaging methods and analysis techniques. And he's been very influential in developing multivariate analysis techniques, such as hyperalignment that he'll be talking to us about today. He has an impressive list of awards in recognition of his work, including NIH Director's Award, career awards from Kavli and Cognitive Neuroscience Society. And he's now arrived at Dartmouth as Professor of Psychological and Brain Sciences and Director of the Dartmouth Brain Imaging Center. And just as importantly as his scientific contributions, he's been an advocate for open science and making methods that he develops and underlying data available. I think almost every neuroimaging tutorial in the 2001 data set, probably the most widely analyzed data set in the field by now. And in support of this, he's also the co-director for the Center of Open Neuroscience, which does some great work getting open source software out. And he's director of the Center for Cognitive Neuroscience at Dartmouth, which has some great summer institutes and training workshops. So, welcome, Jim.
Well, thank you, Roland, and thank you for inviting me. It's a pleasure to be here and to meet you. I've been having some really fascinating conversations this afternoon. What I want to talk to you about is a project that we've been working on for over 10 years now, and that we gave spoiled the English language by giving a new word, hyperalignment, but we wanted something that uniquely talked about this approach that we're using. And this is a method that we've developed for taking the information that's encoded in cortical topographies, which are idiosyncratic. So each individual's brain has a slightly different arrangement of the cortical topography for representing the same information. So this is a method that we have for aligning the information that's embedded in these fine-grained cortical topographies in abstract information space, where the same information is now aligned together. And this is different from trying to align the cortex of one person's cortex and another person's cortex. I hope this makes sense as I go along here. So first I want to just acknowledge the people who actually did a lot of this work, especially Swarup Guntapalli, who was my graduate student and then postdoc. He's now gone truly to the dark side into private industry, a startup company in Silicon Valley called Vicarious. And then the other people have been critical here. I'll be talking about some of Feilong Ma's work on individual differences towards the end of the talk. And then this guy is Yaroslav Holchenko, who is cognitive neuroscientist and software engineer, and a huge advocate of open neuroscience, and the co-developer of these software platforms that are widely used around the world. And then we've been collaborating with engineers from Princeton University, headed by Peter Ramage, who is the chair of electrical engineering at Princeton. He's now the director of their machine learning initiative, and we've burned through a series of graduate students. So this is an outline what I want to talk about today. first I'm going to just try to orient you to what this problem is that we're trying to address, which is understanding how we can model the information that's contained in the fine-grained topographies that are idiosyncratic. They vary from grain to grain. And then the conceptual framework of an information space. This is a high-dimensional feature space, which is how we think about the information that's embedded in these patterns of neural activity. And then how do we derive a model of a shared information space? And then we need transformation matrices so that we can take any individual subject's data space, anatomical data space, and project it into the shared information space, or take information that's in the shared information space and project it into any individual subject's idiosyncratic topography, see what it would look like in that subject's brain. A bunch of validation tests to show that it actually does work. And then I'm going to talk about a few topics that I think are interesting and relevant here. One is how this procedure produces a new model of functional cortical topographies. And I'll go through the math of how that works. And then I'll talk about how, even though the purpose of this project is to find what's shared across brains, it allows us a much more powerful analytic method for studying how brains differ from each other in terms of the information encoded in these fine-scale topographies. And I'll show you some promising initial results that demonstrate that. So this is just an illustration of how multivariate pattern analysis works in sort of its initial manifestation, which is using machine learning pattern classifiers. And for this, we used a support vector machine, that's what SVM stands for. And this is a study that was done by Andy Connolly when he was a postdoc in my laboratory. Some of you know Andy. And subjects looked at 6 different species of animals, 2 species of insect, 2 species of bird, and 2 species of primate. So they saw pictures of these objects while they were in the fMRI scanner. We measured the patterns of brain activity. And then we applied a machine learning pattern classifier to determine whether or not the pattern of activity evoked at each one of these species was unique and allowed us to say what species of animal was the subject looking at when this pattern of brain activity was measured. And this support classifier result is often shown as a confusion matrix. So the input is along the vertical axis, ladybugs, luna moths, mallards, yellow warblers, ring-tailed lemurs, and spider monkeys. And then the classifier output is on the horizontal axis. And if the classifier is working, most of the trials are, the ladybugs are classified as ladybugs, or the luna moths are classified as luna moths. So this diagonal would have bright colors corresponding to high percentage of classification. And the off diagonal, which are all the confusions or errors, should have blue or low errors, low rates of classification. You can see it works really well. And it just gives you an initial look at how powerful this is. So we can tell whether a person's looking at a beetle or a moth, or a duck or a warbler, or even a lemur or a monkey, significantly. So these are fine scale patterns. The problem with this is that this classifier has to be built anew for each individual subject. So we have to take the data from one subject, build a classifier on what's called training data, and then we have left out test data to see whether the classifier is valid. But if we take that classifier built on one subject's data as training data, and then apply it to a different subject's brain data as test data, it doesn't work. And this just shows that. So this is the confusion matrix that I just showed you with the average performance for within subject classification. And this is what between subject classification looks like. So if the person is looking at a duck, it's very rarely classified as a duck trial. And the overall classification actually crashes from 68% down to 37%. So that's the problem that we were confronted with and tried to solve with this algorithm. So first I want to just talk a little bit about what these patterns look like and back up into low early visual cortex to talk about how topographies are structured. So in early visual cortex, this is a map of retinotopic areas starting with V1, and the cortex has been cut along the fundus of the calcarine sulcus. And within V1, there's a topographic map of retinotopic location. So this is a map of the polar angle. And you can see that along the fundus of the calcarine sulcus, there's a representation of retinotopic locations along the horizontal meridian. And then at the border between V1 and V2, ventrally, is the vertical meridian, upper vertical meridian. So there's one cycle of functional topography from the horizontal meridian to the vertical meridian in V1. But there are finer scale topographies as well that are multiplexed with this retinotopic topography. So this is a patch of cortex from V1. This is, map was developed from fMRI data taken with a 7 Tesla magnet at the University of Minnesota, the Koob, and Uberville and company. And this shows the location of ocular dominance columns. So the red patches are pieces of V1 that get input from the right eye, and the blue patches are pieces of V1 cortex that get input from the left eye. And the spatial scale, this is a 1 millimeter spatial scale. So you can see these ocular dominance columns are about 1 millimeter or so across. And so these are all contained within V1 and for different retinotopic locations. And then there's an even finer scale topography in V1, which is orientation selectivity. So this just shows a map of the orientation selectivity columns of cortex in V1. It's the same piece of cortex from the same subject. The black outlines are the outlines of the ocular dominance columns. So if you look at this red ocular dominance column down here, it's outlined here, and you can see that all orientations are represented in a very well organized orientation map within that ocular dominance columns. So these different topographies all coexist in the same patch of cortex, and they're multiplexed to represent the low-level visual features of the visual stimulus. Now, if we move up, we don't know what the basis functions are. So you can think about these topography, retinotopy, ocular dominance, orientation selectivity, color. There are also spatial frequency maps. All those things are basis functions for understanding the functional organization of early visual cortex. We go to higher level visual area. I'm looking here at FFA, which is the fusiform face area. We don't know what the basis functions are. We don't have a good theory of what the different features are, but we know that there are fine scale topographies because we can do decoding of the patterns of response in the FFA. And we can decode things like, is the person looking at a human face or a dog face or a monkey face, even though all three of these evoke strong responses in the FFA, but they're distinct for these different species. You can also decode whether the subject is looking at a full profile or a 3/4 profile or a full face. And also decode whether the person's looking at one identity or another identity. So all this kind of information is encoded within the fine scale patterns in the FFA. And this is all relevant information about what faces look like. But it's even a little weirder than that, because we can also decode whether the person's looking at a monkey or a lemur in the fusiform face area. And we can even decode whether the person's looking at a shoe or a chair. Those are weaker classifications, but that information still exists within this fine scale topography. But we don't know what the basis functions are and how these basis functions are organized at what spatial scale they're organized. So this leads us to the question of this research project. So the first we ask, can the information that's encoded in these fine-grained topographies, including this high-level information, be modeled in a computational framework that's common across brains? So we know that we can't do classification if we just align the data anatomically, but is there some way to somehow align the information that's shared across brains in variable topographies? So our approach is to develop a common high-dimension information space, and then with transformations from the idiosyncratic topographies into the common information space, and then back again, so we can project data from the information space back into the idiosyncratic topographies. So it's important to emphasize here that the common model space is a canonical cortical topography. It's an information space. So we're not trying to find out what is the ideal or central topography across all brains. We study topographies as idiosyncratic instantiations of that information. Okay. So we have a very simple model of this information space. It's just a high-dimensional linear model. I'm sure it's not ideal. I hope it gets replaced with something more powerful at some time, but it works very well at this level of analysis. So the way we think about this is that you can take any pattern of response measured in Cortex. So these are 6 different patterns of activity measured with fMRI. And it's a 2-dimensional array, and you can take that 2-dimensional pattern and vectorize it into a single-dimensional vector of n voxels, or n cortical vertices. And that vector can be thought of as a vector in a high-dimensional space. And the dimensions in this space are the cortical vertices themselves. So this is just meant to represent a 5-dimensional space, V1, V2, then V3, V4, V5, and then that can scale up to very high dimensional. Usually we're talking about 200, 500 dimensional spaces here. And with each new pattern of activity, the vector is in a different location in this vector space. So anatomically defined vector space. Now, so these patterns could be patterns of response to stimuli. This could be the pattern of response to monkeys. This is the response to birds. This is the response to moths. Okay, and then you have these patterns would just be different locations in the vector space. But these patterns can also be patterns of connectivity. And this is a little bit harder to kind of develop intuitions about. But in a patch of cortex, like this patch in lateral occipital cortex, each vertex is going to have a different level of connectivity, strength of connectivity to a cortical target. So the connectivities in this patch are to this cortical target in left frontal cortex. And if the connectivity varies from vertex to vertex within a contiguous cortical patch, it's a pattern. That pattern can be vectorized. So you can express the connectivity data in the same kind of matrix as the response data, where each column is a cortical. Each column is a cortical vertex, and each row is a target, a connectivity target. And so you can think of the patterns of response for this target, and this target as being vectors in a connectivity space. And once the data are in this format, whatever math we develop for this data can be applied to these data. And I'll show you that that works actually really well. So the goal of hyperalignment then is to take these individually idiosyncratic spaces that use the cortical locations as the coordinate axes and find the transformation matrix for each individual subject to project this subject's responses and this subject's responses into a common information space based on the assumption that if they're having the same experience, looking at, for example, the same part of a movie or being in the same experimental experiment, you want to put that together because their brains are representing the same information. So now, in the transform space, the common space, vectors are clustered, are brought together if they have shared information, common information. So to do this, we developed this algorithm called Hyperalignment to calculate these transformation matrices and develop this model of a common or shared information space. So we have people watch Raiders of the Lost Ark, which I know is really dated, but it's still a very entertaining movie. This Harrison Ford is a young man. And so as people watch the movie, we measure their brain activity. So I show this clip from the movie just to remind you that this is not a controlled experiment.
Dr. Jones.
And you can see that, that there's lots going on. It's a very cluttered scene, it's dynamic. There are voices, there's background music, there's a storyline. All of this is providing this rich cognitive space that is part of watching a movie. And these are the patterns of response in two subjects, restricted just to ventral temporal cortex, and they're synchronized. So whatever you see over here is at the same point of time in the movie as whatever you see over here. You can see this isn't a trivial problem, that these patterns don't look a lot like these patterns, but we assume that they're representing the same information. So it works like this. So as the brain produces a new pattern of activity for each time point, you can think of that as a vector in this subject's space, idiosyncratic information space. So these vectors are color-coded so that the blue circles are the same time point, the green squares are the same time point, and so forth. So the challenge is to somehow transform one subject to be in better alignment with the second subject in the representational space. Yeah.
I'm sorry, I'm afraid of the cost, but I don't have the question right now. You're saying, previous slide said that your vector is representing information at that moment in time for each subject. The movie is so sort of the information is so ill-defined, right? And there's visual information, linguistic, auditory.
I would say it's rich, not ill-defined, it's rich. It's all those things.
Okay.
It's all those things.
Okay, so it literally means just everything that they're
Everything that's in the movie is being represented in the brain. When you watch a movie and you appreciate the social context, font, and the suspense, and the music, right? and the scenery, all those experiences are in your brain. Right? And so somehow your brain is where those experiences reside. And so we're taking a very empirical approach. We're not trying to isolate out just the visual stimuli, isolate out just the voices. We're throwing it all in there with this very rich representation. Can we understand how to align the brains with each other? All these different types of information. Yeah.
That you're making the sum for two individuals representing the same.
Right.
So we know, for example,
Yeah.
So I see what you're going to get.
See, you're jumping ahead to the end of the talk, where I talk about how people differ from each other. But the purpose of the project was to find what's common across brains and model that commonality, that information. And then that gives us a lever for understanding better how brains differ from each other. Okay, so I'm sorry, I walked away from the mic and half of you didn't hear me. So we want to derive a transformation matrix that will align subject 2 to subject 1. And we use a transformation called the Procrustes transformation. This is an algorithm with a closed form solution, and it calculates a matrix that's an improper rotation. It's a rigid rotation of the vector space to minimize the differences between the paired vectors. And we realized after we had developed this that we had serendipitously, accidentally made a very good choice, because a rigid rotation preserves the distances between the vectors. So vectors that are very similar are still very similar after a rigid rotation. Vectors that are very far apart are still very far apart after a rigid rotation. And so this shape of similarities and differences among patterns of response is something that's called representational similarity or representational geometry. We know that's highly conserved across brains. And so we're kind of taking advantage of that commonality of representational geometry to make a method that works. So it's a rigid rotation, and this is just an illustration of how you can rotate this pattern of vectors in 3 dimensions. So now the pattern looks a lot like subject 1's pattern. And if you put the vectors from these 2 subjects into the common space, the vectors for the same time points are now close together, whereas before they were far apart. So for example, too late. Now for a 3rd subject who has responses to the same pieces of the movie, this subject's vectors are going to be in very different locations in this subject's space, because these cortical vertices are anatomically aligned with the other subject cortical vertices, but not functionally aligned with their cortical vertices. We have to get a new transformation matrix that's specific to subject 3. And we find that with this subject, we can also rotate his vector cloud, his or her vector cloud, to be in alignment with the average of the first 2, like that. And so we iteratively add subjects and align them to the average vectors from the previous subjects. And then after we go through all the subjects that way, so there's something, the first subject we call the reference subject. But we wander a little bit away from the reference subject because we have a second iteration where we then align even the first subject and all other subjects, average vector locations from all the subjects in the first iteration. We still use the first subject's anatomy to illustrate the results. We could illustrate the result in any subject's anatomy using their transformation matrices, but we use the reference subject's anatomy. Now, connectivity hyperalignment works the same way. Connectivity hyperalignment, we have vectors corresponding to the patterns of connectivity in the cortical area to different targets elsewhere in the brain. And so this is just another way of showing 4 connectivity pattern vectors in subject 1, 4 connectivity pattern vectors to the same targets in subject 2. Yes. In our first paper, we went to the trouble of recalculating everything using every different subject as a reference subject. And we got equivalent results no matter where we started. So it's not sensitive to the, it doesn't seem to be sensitive to the order. So here, and again, we just use the same algorithm to calculate transformation matrices so that these connectivity patterns are now aligned in the common information space for connectivity. And for those of you who need equations, and I ran across some of us who needed equations, it's actually quite simple. This is the basis of this of hyperalignment, which is we have a model space for a cortical area J, and we populate that with vectors, okay, that are the averages of the vectors for individual subjects for time points or connectivity targets, I, multiplied by the transformation matrix. I'm sorry, this is for subject I, multiplied by that subject's transformation matrix. And we derive the transformation matrix to minimize the difference between the transformed brain spaces and the average information space. Now, there's nothing sacrosanct about the Procrustes transformation. It works really well, and we've stuck with it for 10 years now. But there are other ways of calculating these, of deriving these transformation matrices. And our collaborators at Princeton have come up with a couple that work pretty well. One is they use a regularized canonical correlation analysis. Another is that they use a probabilistic method for estimating the weights in the transformation matrix. And they all work, but they usually require that we stay pretty close to a rigid rotation. And there's one more step to make this a whole brain model, and that is that all these algorithms are very agnostic about where information originates and where it's projected to in a cortical field. So if we did it for the whole brain at once, it would probably take some of the information from the occipital lobe and project it into the frontal lobe. And I found that unacceptable. So we developed this locality constraint. So what we do is we do a calculate the hyperalignment model for searchlights of cortical voxels. So that's the disk of cortical vertices around the center. And we get a transformation matrix for that searchlight. And then we move that searchlight one step over, recalculate hyperalignment, get a new transformation matrix. And so we end up with overlapping transformation matrices. And there are about 50,000 such searchlights. And then we aggregate these together into one whole brain transformation matrix simply by, we either average or sometimes we just add weights for voxels being transformed into model dimensions. And so with this whole brain transformation matrix, we can project the data from a single brain into the common model space, or we can take any vector from the common model space and using the transpose of this transformation matrix, project that vector back into a cortical topography.
So this is connectivity, right?
We do it with connectivity and with responsive.
So with connectivity, is the spotlight a target, or is it or does the target sit inside the same spotlight?
Well, the way that in the algorithm, what we do is we use we use searchlights as targets also. So we're hyperlining data in a searchlight, and the connectivities are to other searchlights. And I can get into the nitty-gritty details, but it's so does this work? So the first I just want to show you the math in a different layout. So the matrix B is the brain data of cortical patterns for either time points or connectivity targets across voxels. So each column is a voxel or cortical vertex, and each row is a pattern across those voxels. The transformation, which is what we derive, has the model dimensions, so the coordinate axes for the model space as columns, and cortical vertices, so the anatomical coordinate space, as rows. And these are the weights we're estimating with the Procrustes with hyperalignment. And if you multiply the brain data matrix by the transformation matrix, it takes each pattern of activity, or pattern of connectivity from the brain space and projects it into a location in the information space where the coordinate axes are these dimensions, model dimensions. So this is a pattern of responses across voxels for one time point T2, and it's projected into a pattern of responses across model dimensions for the same time point. And the purpose of this is to cluster these patterns together, or these time points together across subjects. So we should be able to do between subject classification. We can say, if in one subject, this pattern looks like is in the same location in the information space as the responses in other subjects for this part of the movie, he was probably watching that part of the movie when he produced this pattern of activity. So we ran exactly that analysis, and we just used a correlation-based nearest neighbor classifier for 15-second time segments. So we take 15 seconds of brain data in one subject and ask, are the patterns in this subject most similar to patterns of activity in other subjects while they were watching the same part of the movie or some other part of the movie? And we use a sliding time window for all the other parts of the movie. So 1 out of over 1,000 classification. The anatomically aligned data, when we do this by searchlights, only this little piece of primary visual cortex has a classification accuracy that exceeds 5%. After hyperalignment, the classification accuracy increases tremendously, in fact, over 10-fold. And now we start to see that we have classifiable information about what they're experiencing during the movie, not just in early visual cortex, but in higher level visual cortex and ventral temporal cortex, lateral temporal cortex, auditory areas, parietal, and prefrontal. So it's a big effect. And now if we do classification based on all the data in the whole brain, we can classify what part of the movie someone's watching with about 95% accuracy. So just by having someone watch the movie, we can tell with 95% accuracy exactly what part of the movie they're watching at any point in time. Yes? How many what? How many segments? So it's 55 minutes worth of movie data. And we use a sliding window. So we classify one time segment, then we move that over 1 TR, 2.5 seconds, and do that time segment. So pardon me? How many segments? Yeah, so with the sliding time window, we have time segments that are centered at each time point. And so with our current data sets, we have 1,300 time points at half of the movie. I'm sorry, I did it again. So it's a sliding time window. So we have 1,300 for the segments we're classifying, and 1,300 for the segments that could be classified by. If I did a confusion matrix, it would be a 1,300 by 1,300 confusion matrix. Yeah? So the question is, how much data do you need to derive the matrix? So the first point is that we derive the matrix on one half of the movie. For these data, we derive the matrix on one half of the movie, and then we classify the time segments from the other half of the movie. So the data we're classifying is not used for deriving the common model. And then we've done an analysis where we use progressively smaller amounts of data to derive the model. And we find that the more data, the better the performance. But it reaches acceptable levels with about 20 minutes worth of data rather than 55 minutes. Now, another way to analyze the information in the movie is from representation geometry or representation similarity analysis. So what we do, this really uses all the data at once. And we look at the correlation of the pattern of response at one time point with the patterns of response at all other time points. So this is a 13 by 1300, 1300 by 1300 correlation matrix or similarity matrix. And then we ask, is that similarity matrix the same for different subjects? So in auditory cortex, time points where they have the same voice or the same piece of theme music may be quite similar to each other. Whereas a time point with theme music as compared to a time point with loud explosions, those may be very different from each other. So is that kind of similarity structure shared across subjects? So when we look at that, we get a good similarity of this similarity structure, very high, finely grained similarity structure in the anatomically aligned data, especially in visual and auditory cortices. After hyperalignment, that concordance of representational geometry across subjects doubles. So the correlations are almost twice as high after hyperalignment. And this is a surprising result for some, because some people think that if you're looking at a patch of cortex, then it's not so sensitive to misalignment of anatomy. And so it should be that the representational geometry is about the same for a searchlight and the searchlight next to it, and so on. But what this suggests is actually there are fine-grained changes in representational geometry across cortex. And so if you can do a better job of aligning the information across subjects, then you see alignment of these fine-grained differences in representational geometry. Now, another thing nasty reviewers brought up was they said, well, you say it's fine-grained information. We don't believe it's fine-grained. So we did a couple of analyses. I'm showing you one of them here, which is a between subject point spread function of functional topography. So what that means, we take the time series for one time point in one subject, and we correlate that with the time series for other subjects in the same cortical location. So that's a measure of the concordance of function for that cortical location. Then we look at the correlation of the time series for one subject in the cortical location with the time series for adjacent vertices in other subjects. And then we look at that's one step away. Then we look at the correlation of the time series for one vertex in one subject and vertices that are 2 steps away in other subjects. So if that tuning function for a vertex is specific to that vertex, then those correlations are going to go down when you look at neighboring vertices. And you can see in the anatomically aligned data, the time series for adjacent vertices has about as much correlation with a index vertex as the index vertex itself. You really have to get about 3 or 4 steps away before there's a real drop in this correlation. And by contrast with the hyperaligned data, the neighboring vertex has a tuning function that has a much lower correlation with the index vertex than the index vertex in other subjects. So this suggests that we're aligning the functional data at the level of the single voxel, 2.5, 3 millimeters. With connectivity, we see the same kind of thing. So this is the increase in intersubject correlation of connectivity profiles after connectivity hyperalignment of the movie data. And you can see that now we're getting stronger intersubject correlations of connectivity profiles everywhere. including the, these difficult areas like medial prefrontal cortex. Everywhere, we're seeing that there's a very fine-grained variation in cortical, in functional connectivity. So the neighboring vertex is got a connectivity pattern that's not as highly correlated with the index vertex. For the anatomically aligned data, the cortical connectivity looks about the same, 2, 3, and even 4 steps away. And we can, with connectivity hyperalignment, we don't need a movie any longer because we're looking within subject functional connectivity. We don't need some kind of synchronous shared experience. Now we're looking at just shared connectivity patterns. So we can look at that in Human Connectome project, in resting state data. So we pulled some data from the Human Connectome project, ran hyperalignment on it, and got a dramatic increase in the between subject, intersubject correlation of connectivity profiles. And now we can compare that to the within subject correlation of connectivity. So we can calculate connectivities for a given subject from one set of data, 20 minutes of resting state data, and then we can calculate connectivity in the same subject's brain with independent resting state data from other scan sessions. And that's what the within subject correlations look like. And this shows the point spread function for the between subject hyperaligned data in blue and the within subject in green. So you can see that the within subject data shows the same fine-grained variation in connectivity with about the same shape as the between subject after hyperalignment. So we seem to be capturing as much variation in fine-scale connectivity as exists in individual brains. And now we're modeling it in a common space. So that shows you that it works. And now I want to talk about what happened to topography. What are these topographies? I'm talking a lot about topographies, but can we say anything about what they look like? Can we model them? And this framework produces an alternate model for functional cortical topography. And that model starts with the weights in the transformation matrix for model dimensions. Now this is the transpose of the transformation matrix. So now the model dimensions are rows. And these are weights for this model dimension across the vertices of one subject's cortical anatomy. So we can look at that, at this pattern of weights on the surface. Cortical surface is modeled as a triangular mesh. So in an idealized, regularized triangular mesh, we can look at what a pattern for one dimension would be in this cortical triangular mesh. These are different cortical vertices in one subject. And the colors correspond to large weights to small weights. Most of the vertices are blank, 0, because we do a searchlight analysis. So this would be this is just an illustration, of course, idealized illustration of a pattern of weights from a transformation matrix. Now, if you have a different pattern of weights for each model dimension, you can think about that as multiplexed topographic basis functions for these 4 rows in the transformation matrix. And if you find a weighted sum of these multiplexed topographic basis functions, that is the model of the core pattern. And the weights are these y's here. And where do these y's live? Well, the y's are the model data. So these are the values for that pattern in different model dimensions. So if you multiply this vector, let's say it's T2, this vector of a pattern in the information space, by the transpose of the transformation matrix, it creates a topographic pattern in this subject's cortical space. So this is the predicted topography in the brain space. So that pattern, if you multiply that pattern by the column, the transformation matrix, that gives you the estimate for the first cortical vertex in the pattern. You multiply it by the second column, the estimate of the second cortical vertex. Third column, third cortical vertex, and so on, for modeling the full pattern of activity in the cortex. So you can think of that as one voxel, like voxel 3, has different values in each basis function, and you multiply those by the numbers in the model space vector. So this is a cool model. It doesn't have anything to do with topographies we think about, know about. And so what this allows you to do is take a vector from the model space, project it into an individual's anatomy. That vector can be derived from other subjects because everything has been put into this common space. We can have a vector that corresponds to a known topography. First, let's look at what these pattern vectors look like, topographic pattern vectors. So these are 3 topographic pattern vectors in 2 different subjects' brains. You can see they are from searchlights in lateral temporal cortex, and that they look very different for these 2 subjects. It just illustrates the idiosyncratic nature of them. Now, this is where we tried to model retinotopy. So we had 8 subjects that we had retinotopic localizer scans in. So this is the retinotopic localizer for polar angle in one subject. You can see it's a very nice map with borders between V1 and V2 at the location of the vertical meridian, and the border between V2 and V3 at the location of the horizontal meridian. Then we took the other 7 subjects' retinotopic scans, polar angle maps, projected them into the common model space using the transformation matrices that were derived from their responses to the movie. This is why it was kind of a crazy project. We don't make people fixate for the movie. They just look whatever they want to. So they're just watching the movie. They're happy as a clam, enjoying the movie, most of them. But we use the transformation matrices to project those other 7 subjects' polar angle maps into this subject's idiosyncratic anatomy. And then we calculate a polar angle map based on other subjects' data. And that's what this is over here. And you can see it looks almost identical to this. So that model of topography as multiplexed topographic basis functions captures this kind of a topography of retinotopy, polar angle maps. It also captures things like category selective regions and higher order cortex. So in a set of these subjects, we had a standard functional localizer where they looked at still images of faces and still images of objects. So we could find that the vector between the average response to faces and the average response to objects. And using the movie data derived transformation matrices, we projected that vector into the common model space. So we had an average face versus object vector in the common model space based on the localizer data. And then we can project that based on other subjects' data into a left out subject's cortical anatomy using the transpose of that subject's transformation. And so this is the result for two subjects. So the location from that subject's own localizer data is shown in outline. Yellow around the FFA, green around the PPA. Fusiform face area, perihippocampal plates area. And the map of colors shows the topography of that face versus object vector in the subject's anatomy. You can see it maps remarkably well onto the individually defined FFA. But these maps are from other subjects' localizers. And it's even capturing the idiosyncratic shape of the FFA in each subject. So this FFA has a quite different shape from the other subject's FFA. But the mapping the same vector from the common model space, virtually the same vector, from other subjects into their anatomies picks up the details of how the FFA is configured in the individual anatomy. Now this says, so okay, so these things exist in the common model space. If you find the right vector for a polar angle map, or the right vector for face versus object contrast, it does a very good job predicting the topography, idiosyncratic topographies for those contrasted individual grains. So where is that in the common model dimensions? Is it that one of those dimensions is a polar angle dimension, or is one of the dimensions, those multiplex topographic basis functions, is that the FFA? And the weights for everything else is 0. Well, it doesn't work that way. And this just shows the topographies for 4 topographic basis functions, but these are no basis functions for principal components. So they're principal component analysis. So the PC1 is the one that accounts for the largest increment of variance and variation of the patterns of response in ventral temporal cortex. This accounts for the second largest increment of variance. And you can see that none of these really picks out the FFA in its entirety. PC1, curiously, picks out little tiny pieces of the FFA, the whole thing. And then the FFA really is just not identifiable in these other ones that are accounting for a lot of variance. So it's really, you have to multiplex the basis functions to recreate these topographies. So when we look at how much variance is accounted for by PC1, PC2, PC3, et cetera, we explain about 50, I think it was 55% of the variance overall, to go out to 35 principal components. If we look at how, just the first principal component accounts for 22%. The first 3 accounts for about over 30%. The face versus object vector in ventral temporal cortex accounts for about 7 or 8%. So that topographic feature of ventral temporal cortex, which exists, which is a very important feature, is just one, is one relatively small component of explaining the topography of ventral temporal cortex. So I've got lots of ideas about why that's the case, but I'm going to skip ahead in the interest of time to talk about individual differences. So this is work being done by Feilong Ma, and his family name is Ma, but when he published his first paper, we had a discussion. He said, what do you want your name to be for the scientific record? There are millions of Ma's right out there. Feilong is a much more unique name. And outside of China, no one can tell the difference between the family name and the specific name. So he's now is Feilong in the literature. And what he did is he developed a way of looking at similarity between pairs of subjects either in their patterns of response or their patterns of connectivity. So we would take the time series for all the cortical vertices in a searchlight and vectorize that into one-dimensional vectors, so we could correlate the patterns of response across voxels in one subject to the patterns of response across the same voxels in a second subject. And that's an index of how similar they are in the way they responded to the movie. And so in the anatomically aligned data, these correlations tend to be fairly low, 0.2 to 0.3. But you put these then into a matrix of the differences between subjects. So this is an individual differences matrix. So each square in this is how similar one subject is to another subject. So the whole matrix is all the possible pairwise similarities across subjects. So the first question he asked is this individual differences matrix a reliable object? So he said, if we derive that individual differences matrix for the same brains again, but from independent data, like for moving, from a different set data set, we get the same pattern of who's alike and who's different. And they're remarkably similar. The similarity is up around 0.77 for this particular cortical area. So then he did the same thing for hyperaligned data. So for a searchlight in the reference subject's brain space, he could look at the vectorized response across model dimensions and see how similar two subjects are to each other. And as expected, subjects are much more similar to each other. That's the whole purpose of hyperalignment. And so the individual differences matrices have higher values. So then the question is, well, is this reliable? We just removed a lot of what makes subject brains different from each other. And there's nothing about the procedure that removes noise. So we, of course, expected that the correlations would be lower between the different individual. But actually, they're higher. So this just illustrates it. This is a map of the reliability of individual differences matrices across the brain in anatomically aligned data. You can see we get variable measures of the individual differences in the auditory, visual, parietal, and prefrontal areas. Very low reliabilities in the sensory motor areas because these people are lying on the scanner doing nothing. They're just watching a movie. So the patterns of activity and in those areas are variable for idiosyncratic reasons. Okay? Idiosyncratic within subject across sessions. After hyperalignment, these reliabilities go up dramatically. And now we see reliabilities, strong reliabilities in interesting places like medial prefrontal cortex that plays a big role in social cognition. But we scratched our heads and we said, how could this be? Why is it more reliable after hyperalignment? So Fei Long did something very clever. He took the data for each searchlight and he decomposed it into a coarse spatial scale component and a fine spatial scale component. So for the coarse spatial scale component, he just looked at the average time series or the average pattern of connectivity across all the vertices within that searchlight. And then you can look at how similar people are in the average response for a searchlight. Then he took the time series for each vertex and subtracted from that the average time series. And so now the time series vary vertex by vertex around a mean of 0. And when he did that, the coarse scale, he found that the reliabilities for the anatomically aligned data and the hyperaligned data are essentially the same. So hyperalignment does nothing to align individual differences based on coarse scale function. When you look at the fine scale, he found that the reliabilities went down a little bit for the anatomically aligned data because it's doing a very poor job of aligning the information in those fine scale topographies. For the hyperaligned data, it greatly increased the reliability of the individual differences. And this scatter plot shows that the reliabilities for the hyperaligned data are consistently higher and the reliabilities for the anatomically alignment. So each dot is a searchlight. You can see almost all the dots are above the diagonal. So what this seems to be doing is that by removing individual differences that are due to idiosyncrasies of cortical topography, it allows us to see more clearly the underlying and the information that's encoded in this cortical. And we know that that fine scale information is where interesting things reside, like the fine scale distinctions between visual stimuli and whatever is going on in frontal and parietal cortices. So then we asked is, does this mean anything? So we have reliable individuals. Is it related to anything? So we analyzed data from the Human Connectome project, and from that he got a resting state data for 1,002 brains. And he hyperaligned all these data using connectivity hyperalignment. And he derived a template of a common model connectome from independent data, and then aligned all 1,000 brains to this template. And then for each searchlight, he predicted G, general intelligence. So with the Human Connectome project, they have test scores for tests these people take. Things like picture vocabulary, progressive matrices. And so he found that he could predict general intelligence from the patterns of connectivity in the data that's aligned mostly anatomically using the Human Connectome project MSMAll, which is for multimodal surface chain, particularly in parietal cortex. With the hyperaligned data, those predictions are much, much stronger. So whatever the cortical basis is for intelligence, it seems to reside largely in what is in fine scale structure, fine scale topography. And the only way we can really look at the individual differences is by doing something like hyperalignment to clear away the weeds that are, obscuring that of individual differences in cortical anatomy. Okay. So I've talked longer than I should have. I have another section on how you can use representational geometry to model how information is transformed along processing pathways. But I'm going to skip it in the interest of time to give you people some time to ask more questions. So let me go skip ahead across all this to just as the major elements of the common model derived with hyperalignment. So the basic component is there is this common high-dimensional space, and you can have information about representational patterns or about connectivity patterns. And then there are individual transformation matrices for each individual subject. They have to be derived individually for each subject. And we have this algorithm that we've developed for calculating these transformation matrices. And by getting transformation matrices for a group of subjects, we can project all those subjects' data into a common space, which gives us this a common space with average vectors for connectivity or response. The distances between pattern vectors that contain the same information are minimized. That's the whole purpose of hyperalignment. And because we use Proquestes or something related, preserve the vector geometry from the individual brains into the common model space. Common model dimensions are very complicated things. They have lots of information loaded into them. Each common model dimension is associated with a response tuning function, and these response tuning functions are common across brains. And they're valid for diverse domains of information. So it's not just visual cortex, it's not just visual perception, it's also in auditory areas. It's also in places like parietal cortex and prefrontal cortex. Each one also has a functional connectivity vector that's common across brains. And now these functional connectivity vectors that are common across brains include everything, including the medial prefrontal cortex. And both of these, the tuning functions and the functional connectivity vectors, are capturing variations with a very fine spatial granularity, a single voxel. And then each common model dimension also has topographic basis functions that are specific to each individual. And these reside in the transformation matrix. And they can be used to model individual variations in functional topography. And then what we've been discovering in the last couple of years with Phelan is that this also provides us a new approach to understand individual differences. Because individual differences in the common model capture how the representation of fine scale information varies across brains. So it increases the reliability of individual differences, better predicts cognitive differences, which leads to the conclusion that perhaps the granularity of cortical architecture that underlies cognitive differences is matched better by the scale of fine scale topography than by the scale of coarse scale topography. So just reminders that the common model space is not a canonical cortical topography. Rather, the cortical topography is idiosyncratic to each brain. Each brain is an idiosyncratic instantiation of how that information can be encoded in an individual brain. And then I want to finish with this slide just to plug the work of Yaroslav Halchenko and Michael Hanke, who are the developers of these wonderful software packages. All the software for hyperalignment is on the MVPA platform. I think you people use NeuroDebian here, right? Yarek is the director of the Center for Open Neuroscience of Dartmouth. They're both huge advocates of open neuroscience, free and open source software, openly shared data with no strings attached, and open instructional materials. And then we have 3 papers. The hyperalignment papers by Swarup and Papali are these 2. And then Phelan's initial paper on reliability of individual differences is in neuroimage. And he's going to present the data on predict intelligence at the Organization for Human Brain Mapping this summer in Rome. It's a great opportunity for a junket. And that's what I want to say. Thank you very much for your attention. Yes.
What I'm showing here is that the patterns show subjects processing the same type of but on the side of the information itself, you think that this
It's on.
Does that work?
Yes, it does.
It's work. Now I can walk.
With movies, I mean, the text would be pretty complex, so I have to combine something similar. Speech tell us something about, say, this thing, this type, the gender of this type of speech. Right. Can we gain information from this kind of data?
So our approach has been not to worry. Sure. Information rich there is. But there are ways of starting to ask what type of information we could and that falls into picture model in theory, you should be able to then predict the brain response in some sense. The Okay. They use a deep neural network to project, to create feature spaces with the dynamic features. Develop the dark features. They show that the group process with Hyperalignment, my colleagues, data from chapter. They didn't collect any data. Now, when I look at that, though, I said, gee, that doesn't look like it. And we did another analysis. Movement. Nature. And so we got data saying kind of animals and what they do, what's the background to them. And so putting the biggest responsibility based on the information. We found that the cooking process prediction based on the stimulus, it was improved by hyper. It wasn't improved enough of that structure. And so what that leads me to is somehow the information, the really lot of information doesn't have to be so it's a problem. How do we model these technologies? What's in the stimulus? What's in the scene? We're still stuck in that model with a single word label. It's very often very sensitive how that falls short. What we're really good in this. And so I think that it should work. We need to based on what? Based on very smart people. Yeah.
Nice, just actually we can pass the mic.
I had a clarification question and then also just a larger sort of conceptual question. First of all, really enjoyed the talk. Super, super interesting. I'm curious about, so you talked about having essentially taking data from a bunch of subjects and hyperaligning them and being able to predict the multivoxel pattern of a left-hand subject.
Right.
And the thing that I'm trying to just make sure I understand is what exactly is needed to do that transformation to the left-hand subject. You need some kind of, you need the transpose.
You need the transformation matrix.
Right. So,
and you have to have data for that subject on, that allows you to calculate the transformation for that subject's brain space to the templates.
right. So
And before, we thought, well, they have to watch the same movie. And so I would give this talk in earlier versions, and people would say, gee, that's really interesting, but I'm not going to waste an hour of my fMRI time to watch people with Raiders of the Lost Ark. But now that we have connectivity hyperalignment, it works on the resting state data. So everybody's collecting some resting state data. I don't think it works as well, but it works acceptably well. And we're also finding that we can hyperalign based on connectivity. If somebody watches their favorite movie rather than Raiders of the Lost Ark, like Grand Budapest Hotel, right? Then we can take that person's connectivity while they're watching the Grand Budapest Hotel and derive a transformation matrix to a template based on Raiders of the Lost Ark.
got it. So the data don't have to be from that same task, but you do need something to figure out that transformation.
You just have to have some way of figuring out that transformation. And now with connectivity hyperalignment, it gives us a lot more freedom about what that could be.
Very cool. And then the conceptual question was, so one goal that a researcher might have is to figure out which brain regions contribute largely to a classification.
Right.
And so one sort of, maybe not hesitation, but one question that I have is, when you put everything into informational space as opposed to anatomical space, does that limit in some way the amount of generalization we can do, or the conclusions we can draw at the group level about which particular brain regions, anatomical regions, support particular functions?
Yeah. Well, to say which brain region, you're back into an anatomical space, which means you have to project your classification results into individual subjects' anatomy, and then do the statistics on the anatomical data about classification accuracy.
Okay, cool. That
Yeah.
makes sense. Thank you.
I'm glad you ended up on the course versus mine distinction. I'm glad that you ended up on the course versus mine distinction, because I think your original statement that between subject classification doesn't work, not quite right, because early stuff by Tom Mitchell and others did in fact try to do between subject classification.
Right.
It worked statistically significantly above chance.
It worked pretty well.
Yeah.
And I can tell you why.
Okay. So does it work well for the same reason? That is that you're looking at the coarser scale, and in limit, that coarser scale is going to align itself better than the fine scale.
Yeah, part of it is that if you're looking at a coarse distinction, you can do it with anatomically aligned data. So if you want to classify responses to faces as compared to responses to houses, you don't really need anatomical alignment to do that. The other thing about the way Tom Mitchell did his work is he really looked at the whole brain. So he was looking at whole brain patterns. So these are coarse patterns that are more just, more easily classifiable rather than the pattern within a cortical patch. So when we do our classification of movie time segments, if we do it for the whole brain, okay, where then we could take advantage of the fact that the face area is a long way away from the place area, auditory areas are a long way away from visual areas. The classification accuracy for the whole brain goes up from less than 5% for searchlights to about 75% for the whole brain.
This would be similar to 5 months.
Yeah, and Marvin Chun's connectome is based on connections between whole brain, connections between cortical areas, rather than connections on a vertex by vertex basis. So there's a lot of information in that coarse scale whole brain connectome, or the coarse scale whole brain pattern of activity that that you can harvest with anatomically aligned data and get really cool results. But so for the movie time segment classification, it's 75% with anatomically aligned data. We're up to 95% with hyperaligned data. If you care about that, you think 95% is better than 75%. If you just want to say, oh, the information's there, well, the information is there in the anatomically aligned data. It's just different information.
So, a completely different question is, how far do you push hyperalignment when you're looking at brains of different sizes?
Boy, that's something we want to do. We haven't done that yet. I think we're going to look at, there's some really nice databases that are starting to be available, like the ECD database, Baby Connectome database, so we can start to look at what is the similarity between children of different ages. And then the CamCam database from Cambridge has data from ages 18 to 88, so we can also see how these things change across the lifespan. We haven't done that yet, but I suspect that it's going to be very interesting. Yeah.
Walk this microphone over to Jane. In the meantime, I'm going to ask you a question. The individual differences analysis with the IQ data, was that on pattern analysis or vocabulary or full scale IQ?
It was a weighted sum of crystallized intelligence. It's mostly crystallized. Yeah.
So one way to imagine that what you see in the dimensional differences is you mapping people to common informational space, and what you're getting out of it is essentially continuing how well they are aligned to that space. Right? Mm-hmm. So it's essentially you have just a gradient of goodness. Imagine cases where where we operate in sort of systematically different informational spaces that are similar, but they're different, right? So we have one group is sort of a little bit different.
Right.
And so can this pick up that?
Yeah, so we're reanalyzing the data set right now from Uri Hassan. He had a really cool paper. The first author is Yashurin, where they had subjects listening to a story. It's a story by J.D. Salinger called Pretty Mouth and Green My Eyes. And it's a very ambiguous story about a late night phone conversation between two men. And it's unclear what they're really thinking and what is really going on. So what they do is they give different framing context before they hear the story. So for one group of subjects, they say, this is about a man who's worried that his wife is cheating on him. Right? And the other one says, whose wife is cheating on him. The other context is, this is about a man who's paranoid, okay? And so your interpretation of what the people are thinking is really colored, is determined by this context. And so, so we have two groups of subjects. We have a group of subjects who see the story one way and a group of subjects who see the story the other way. And with this, you can distinguish it. They distinguish it with these two groups without hyperalignment. We're finding that with hyperalignment, we're getting much stronger distinctions between the groups, including medial prefrontal cortex. And so, yeah, I think that it should be possible now to start to, we could cluster subjects. We could say, we could have the Republicans and the Democrats, or what.
So in this case, the clustering is sort of by design, right? So this group and that group.
Right.
Are you doing sort of separate analyses?
We haven't done that yet, but that,
But that's the idea, right?
But that you should be able to do that. Say, that we've got a group of subjects who have similar brain connectivity patterns. We don't know why they're similar, but here they are. And then so you could do that. Can just speak up.
So connectivity-based hyperalignment, are you first doing registration to template and define that using hyperalignment?
Okay, this is so what we did was first we identified the connectivity targets in anatomically aligned cortical surfaces. And there are searchlights. And instead of just looking at the average time series for the searchlight connectivity target, you decomposed into three principal components. We picked up some of the structure within that searchlight. And then I kept hammering Swaroop to do a second iteration. So I thought, after you do the hyperalignment once, now the connectivity targets have moved because of hyperalignment. And so we should do it again. We should have a better estimate of the connectome. And he did it, and it helped a little bit, but it wasn't enough. And I couldn't motivate him to keep working on that problem. So the way it's working right now is with the searchlights placed on anatomically aligned cortical surfaces.
So are you introducing some bias in which regions you're going to attack things like variability? So for example, the IVP is not more
right.
And I noticed that you're really strong work performance
Yeah.
So is there some type of systematic bias?
So there's clearly a gradient of the size of intersubject correlation of response profiles or representational geometry for the movie. And it's strongest in the sensory motor areas. And it's weaker in things like prefrontal cortex, although we are aligning something there. So the question is, why is that? Is that because there are bigger intersubject variations in the anatomy there that we haven't aligned yet? Or because people have more subjective variability in how their inferior frontal cortex represents the movie? So when we look at connectivity, then the connectivity pattern can be aligned with equal efficiency essentially everywhere. And the only thing that limits it seems to be imaging artifact.
So this is a kind of follow-up. The way I understood it to begin with was that it would straight in that alignment is that my similarity space should be very similar to Roland's, and using that to essentially then transform and do the rotation to initially Roland's space and then you're averaging.
Right.
But if I understood your answer to Roland's first question, it sounds like there's a second constraint that's also built in, which is not just that we have similarity space, but that's one constraint. The other constraint is that there is still an anatomical constraint built in by the way in which you are.
Yeah, I mean, we're doing our best to factor the anatomical constraint out. But I'm sure there's some residue from it, right?
Do you have any sense of how strong that constraint is?
No.
I mean, beyond that.
Not really. Another thing that we want to do that we haven't done yet is look at individual differences in the transfer matrices. So right now we're looking at individual differences in the information after we factor out as much anatomical variability as we can. But the transformation matrices should be measures of how much the anatomy has to, functional anatomy has to be moved around and in what ways it moved around. And are some subjects similar in terms of, how their anatomy differs from, how it differs from other subjects' anatomy. So, would we be able to find, transformation matrices that are different for people with small FFAs as compared to big FFAs? We know V1 cortex can be 2 times bigger than other subjects. Would we see that? These boundaries move around. Would we be able to align the cortical anatomy better, or find individual differences in the cortical anatomy? And is there information there? Is it an apocryphal story that people who play violin have a larger representation of their left hand than apocryphal? Is that real? I think it's real. I mean, that, if you do something for 4 hours a day, it requires very fine motor control of your left hand. Your sensory motor cortex grows, right? And so that would be a meaningful difference associated with some behavioral difference in cortical topography.
The question's here. I wanted to go back to the issue of the rigidity of the transformations that you're performing, and you mentioned that that's preserving certain distances within the informations, many-dimensional information space. But I wondered if you could tell us a bit more about what that means.
Well, that's the representational geometry, and so that shows that some vectors, some patterns are very similar to each other, where the pattern of response to two faces can be very similar, right? And some patterns are very different. So a pattern of response with a lot of faces on the screen as compared to when you're looking at the ocean and a submarine are very different. And so that's what it's.
So, okay,
Okay.
So we should wrap up here. So thank you very much, Jim.
Okay, thank you.